Welcome to Respiratory System Development, Part 1, the Conducting Zone. The respiratory system provides for exchange of oxygen and carbon dioxide to and from the blood. Respiratory organs include the lungs and a branching system of tubes that link the sites of gas exchange with the external environment. Now, anatomically, the respiratory system can be divided into the upper and lower respiratory tracts. Histologically and functionally, the system has two components: the conducting portion or zone and the respiratory portion or zone. In previous videos, I've covered the histology of the respiratory system, but this tutorial covers the development of the conducting zone. I'm Dr. Catherine Moore, the histology wizard. Now, the upper respiratory tract consists primarily of the nasal cavities, paranasal sinuses, and the nasopharynx, while the lower respiratory tract consists of the larynx and the trachea, and within the lungs, the bronchi, bronchioles, and alveoli. Recall that there are changing functions as we move from the upper respiratory tract to the lower, from purification and protection to detoxification and gas exchange. And development is going to reflect those changing functions. So development of the respiratory system starts at about 4 weeks of development, but it continues even after birth. Now, it's important to note that the development of organ systems are connected, and the development of the esophagus and the stomach and the trachea and lungs are linked because they all arise from the foregut. The developing kidneys will produce fetal urine at about 16 weeks, which contributes to amniotic fluid, and that amniotic fluid is critical for lung development. So in terms of anatomy, understanding these shared embryonic origins can help actually make sense of the adult anatomy. In terms of congenital abnormalities, this means that defects in development of the foregut can affect both the upper GI tract and the respiratory system. And it means that kidney defects can also affect lung development. Finally, if we understand how the lungs develop normally, this can actually help clinicians, since management of insufficient lung development depends a lot on the stage. Now, as we move through respiratory development, try to predict what happens when each process we're talking about goes wrong. This will help you so that when you see a congenital abnormality, you can better understand what went wrong with the development to result in that problem. Now let's start first by looking at a timeline. The lower respiratory organs begin to form during week 4. Now remember, by this point that flat trilaminar disc has formed during gastrulation and body folding has begun. The first step in respiratory development is the formation of the respiratory diverticulum off of the foregut. And by the time body folding is complete, the primary bronchi have formed. So at this point, we now have separation of the upper GI system. In other words, we've separated the esophagus from the respiratory system. As the primordial lungs continue to undergo branching, the growing lungs will start to carve through the pericardial peritoneal canals, forming the definitive pleural cavity. Now, at about 16 to 17 weeks of development, terminal bronchioles will begin to form, and the conducting zone is essentially formed by 17 weeks. But development of the gas exchange apparatus, or the respiratory zone, continues until around 8 years of age. Now let's look at a timeline, but in a slightly different way. So in this cartoon, you see the timeline along the bottom, and lung development divided into 5 named stages: the embryonic stage, the pseudoglandular stage, the canalicular stage, the saccular or terminal sac stage, and the alveolar stage, with that continued development postnatally. Now we're going to come back to these different stages several times during this video and the next as I discuss respiratory development. And it's also important to learn these 5 stages because they are definitely on the boards. Now, to simplify things, I really like to think of the respiratory system as a tree. The conducting system, the larynx, the trachea, and the bronchi, all the way to the terminal bronchioles, are like the trunk and branches of this tree, and the alveoli are essentially the leaves, performing the business of gas exchange. So in respiratory development or complete lung formation, there are 2 major developmental processes. The first, building the tree, or the conducting zone, involves a process called branching morphogenesis, while alveolar differentiation, or formation of that respiratory zone, involves the formation and differentiation of this specialized unit for gas exchange. In this video, I'll cover building the tree. So how do we build that tree? Well, we're going to form a single foregut tube, separate it into the esophagus and trachea, and then subdivide that trachea into two bronchi forming two lungs. Then undergo branching morphogenesis to further subdivide the two lungs into lung lobes, segmented bronchi, and the bronchioles. Now, all of these events again occur by roughly 17 weeks of development. And this is essentially the equivalent of the embryonic and pseudoglandular stages that I just talked about. So we're going to start at the top with formation of the foregut. So here you see two cartoons, both sagittal sections of embryos, both showing the primitive gut in yellow, which extends from the oropharyngeal membrane to the cloacal membrane, so essentially the primitive mouth to the primitive anus. Now the foregut extends from the oropharyngeal membrane to the liver diverticulum. Now I've used the word diverticulum a couple times, and essentially diverticulum means that this is a blind tube or evagination that leads from a cavity or passage. Now the most cranial part of the foregut is the pharynx, and the pharynx extends from the oropharyngeal membrane to the respiratory diverticulum that you see here in blue. Lung development then starts with that evagination of the foregut, a pouch that grows out called the respiratory diverticulum, or sometimes the lung bud, and again, it's here in blue. The opening of the respiratory diverticulum into the foregut is the laryngeal opening, and that's going to be the site of the future epiglottis. Now let's take a closer look at this process. Here's a cartoon showing the pharynx, so the foregut in yellow, surrounded by visceral or splanchnic mesoderm in pink, and I'll use these colors for the next several slides. Now you can see that the respiratory diverticulum has evaginated out to the side, and the laryngeal opening I've put on here in blue. Now if we take a section through this tube right at the diverticulum and look at the cross section, you can see the pharynx on the left and that diverticulum, which we now call the laryngotracheal tube, on the right. And here's the same thing from a slightly different perspective. So at this point, we have a tube with the pharynx leading into what will become the esophagus and another part of the tube that will form the larynx and the trachea, but they're still connected. So the very next step is the formation of a wall or a septum that will give us two separate tubes to keep our food and air separated. Now let's see how that happens. So we're looking again at a cross section with the pharynx and the laryngeal tracheal tube. Now two sides of that tube begin to grow more towards one another, and they form the tracheoesophageal folds. The folds will continue to fuse, and the diverticulum will continue to grow downwards, and you can see the forming septum and the separation of the esophagus and the trachea. Now eventually those folds will fuse completely to form the tracheoesophageal septum. Now if we look a little bit later and we look at a cross section, you can see the completely separated tubes, the esophagus and the laryngeotracheal tube. Now eventually in the adult, this ends up as a trachea and the esophagus separated by connective tissue. So that tracheoesophageal septum becomes the connective tissue between the trachea and the esophagus. All right, so now we have a separate esophagus and a separate trachea. Now let's take a quick look at the tissues that make up the trachea. First, let's take a cross section and look at the trachea as a tube. And this very simple cartoon shows the inner endoderm in yellow, surrounded by the visceral mesoderm in pink. Now, the endoderm is going to give rise to the epithelium and glands of the tracheal and pulmonary epithelium, and the visceral mesoderm is going to give rise to the cartilage, smooth muscle, and connective tissue of the tracheal wall and the bronchial walls as well. Now here on the left, you see a cross-section showing those endodermal and mesodermal derivatives in the adult trachea and the actual cross-section on the right. Now I want to recap where we are now. So now we've formed a single gut tube and divided it into the esophagus and the trachea. But what happens if the foregut fails to divide properly? In other words, what happens if the esophagus and the trachea fail to separate in the first place? Well, this can result in a set of congenital anomalies known as esophageal atresia and/or tracheoesophageal fistula. Now, first of all, I just want to mention that atresia means the absence or complete narrowing of an opening or passage in the body. And you can also have a duodenal atresia, jejunal atresia, etc. Normally, the tracheoesophageal folds fuse to form the tracheoesophageal septum. Again, that separates the trachea and the esophagus, just like you see here. But if these folds fail to fuse completely, it results in incomplete division of the cranial foregut into the esophagus and trachea. So in this cartoon, we see the most common result of this failure, which is a tracheoesophageal fistula, or an abnormal connection between the trachea and esophagus, combined with esophageal atresia, which is a blind ending of the esophagus. Now, there can be different results of this failure of the septum to form, and a few of the different anomalies are shown here. You can also have isolated atresia, but that usually is a result of a failure of esophageal endoderm to proliferate. Now, looking at these images of these different anomalies, what kinds of symptoms would you predict from this embryological error? Well, one thing might be aspiration of food into the lungs and air into the digestive system, and this often occurs. Also, that blind end of the esophageal atresia can prevent swallowing of amniotic fluid and prevent it from returning to the mother. This causes polyhydramnios, or too much amniotic fluid, which can cause a lot of different issues. There also can be problems swallowing and feeding. Now, while we don't know precisely how this defect occurs, it is fairly common since it occurs in 1 out of 3,000 live births. It's often associated with cardiovascular or other abnormalities, and it's actually part of the ventral disorder. Now, here in this X-ray from an infant diagnosed with tracheoesophageal fistula and esophageal atresia, you can see a nasogastric tube that's coiled in that blind ending esophagus, and you can also see air in the stomach. All right, now we have a separate trachea, so we've really built that trunk of the tree, but we really need more branches. We need bronchi and bronchioles that will conduct air to the base of the lungs. And like tree branches, these bronchi form by forming buds. So what happens next is that the respiratory diverticulum is going to bifurcate into 2 primary bronchial buds. And as these buds grow and branch and divide, they're actually going to grow into those pericardial peritoneal canals, and they're going to push the pluriperitoneal membranes to fuse together. So what this does is to close off the 2 pleural cavities and make them separate from the pericardial cavity. Now I want to look a little bit more in detail at the lung buds. So just as the tracheal endoderm was surrounded by visceral mesoderm, it turns out that these lung buds are also ensheathed in visceral mesoderm. Now remember from gastrulation that lateral mesoderm splits into somatic and splanchnic components, or parietal and visceral components, and the splanchnic component forms serous membranes surrounding viscera. More laterally, you'll see that parietal or somatic mesoderm. Now as the lung buds continue to grow and branch, carving into the pleural cavity, or space, the visceral mesoderm is going to form the visceral pleura that is directly adhered to the outer surface of each lung. And the parietal mesoderm is going to form parietal pleura that lines the wall of the thoracic cavity. Now importantly, these 2 layers are not entirely separate. The parietal pleura is contiguous with the visceral pleura at the hilum, where pulmonary vessels and bronchi and nerves enter the lungs. You can kind of see that, I think, a little bit better in this cartoon. You might want to think about the location of the parietal and visceral pleura by thinking of pushing your fist into an underinflated balloon. So your fist is going to represent the developing lung, and inside the balloon is the pleural cavity. The balloon itself is the pleura, and that space inside the balloon, again, is the cavity, which will become the pleural cavity. As you push your fist into the balloon, it becomes almost entirely covered in the balloon. So this balloon, or pleura, covering your fist, is the visceral pleura. And all that rest of it, the pleura that's not covering your fist, would be the parietal pleura. But they're continuous with each other. Now that space between the parietal and visceral pleura is the pleural cavity. And in between is a layer of serous fluid produced by mesothelial cells that reduce friction and surface tension between the lung and the thoracic wall. Now, you might remember that mesothelial cells are thin squamous cells derived from mesoderm, and they line inner surfaces. In this case, they line the lungs, and they secrete that serous fluid. Now, a couple of really important points about the pleural cavity. First is that the two cavities, as you can see here, are completely independent, which helps contain the spread of infection. It also means, for the most part, the function of one lung isn't compromised by the other. Now, the second thing to note, and it's really not obvious from this picture, is that the lung almost entirely fills the space in the thorax. And you can really see that a lot better when you look at the adult lungs. Now, third, the pleural cavity isn't this big, large space, but it's actually what we call a potential space between the visceral and parietal pleura. What does this actually mean, a potential space? Well, it really means that these two adjacent layers are normally so compressed together, or what we call opposed to one another. They just have that little fluid layer in between. You can really see how small the space is in the cartoon of the adult lung. Now, what do we know about how these lung buds form? Well, it turns out that the endoderm and the mesoderm are really important for forming the lung buds in the first place. The two tissues need to interact, and the visceral mesoderm releases molecular signals that cause the endoderm to form a lung bud. Now, this process is called induction, and it happens in multiple body tissues during development, and we'll talk about a lot more of these events. Now, interestingly, this ability isn't specific to visceral mesoderm. Turns out that other kinds of mesoderm from branching organs like the salivary glands can induce lung buds in mouse models. But it's actually the endoderm that determines what kind of epithelium differentiates. So we can take salivary gland mesoderm, and it will induce lung if we put it with lung endoderm, but salivary gland with salivary ectoderm will give rise to salivary buds. Now, we don't know the exact signals in humans, but in mice we know that this process requires retinoic acid. So what would you predict might happen with loss of retinoic acid signaling, or maybe a vitamin A deficiency? Well, in mice, this results in pulmonary agenesis or hypoplasia. So pulmonary agenesis and hypoplasia, so no lung or missing lung or a very small lung, are rare occurrences in human, and this is probably because bilateral agenesis is simply incompatible with life. But here you can see an X-ray from a neonate showing left unilateral pulmonary agenesis. So there's no left main bronchus or other airways or even vessels on the left side. Now, interestingly, if there are no other associated anomalies, sometimes diagnosis of unilateral pulmonary agenesis is greatly delayed. So it's often made in adulthood in asymptomatic patients. In fact, one of my clinician colleagues had a patient whose lung agenesis was discovered in a chest X-ray when she was in her 90s, which is just crazy. All right, let's recap again. We've gone from one tube to a separate trachea and esophagus. We've formed two lungs. Now, let's look a bit closer at the development of the bronchi and bronchioles, with those first three branching events being very stereotypical. So starting with those bronchial buds, each is going to differentiate into a primary bronchus, which runs into the hilum of the lung. Now here's where the two lungs begin to look slightly different. So the right bronchus is slightly wider, ultimately shorter, and it's more vertically oriented than the left primary bronchus. The angle that the right bronchus leaves the trachea at is about 22 degrees, while the left is about 36 degrees. And this is why, in the adult lung, aspirated foreign bodies are much more likely to lodge in the right bronchus or branches. It's all due to the embryology. Now, after that initial bifurcation, two buds of the secondary bronchi form on the left and three on the right, setting up the 5 lobes of the adult lung. Now, if we move forward a bit in time, we can see that on the right, we've formed the superior, the middle, and the inferior lobes of the adult lung, while on the left, we've formed the superior and inferior lobes. Now, moving forward in time again, each lobar bronchus is going to undergo progressive branching to form segmental and intrasegmental branches. So there's roughly 10 tertiary or segmental bronchi that form in the right lung and about 8 in the left, establishing those bronchopulmonary segments of the adult lung. Now, the contributions of the endoderm in yellow and the visceral mesoderm in pink are the same as we've seen in the trachea and the larger bronchi. So this branching is going to continue, and it's going to progressively form smaller and smaller branches all the way down to terminal and respiratory bronchioles. This happens over time in about 24 weeks. Now, I think this branching is one of the coolest processes in the body, and it's actually called branching morphogenesis. Now, branching morphogenesis builds the ramified epithelial trees of the multiple organs in the body. So this includes the collecting ducts of the kidney, which we'll talk about soon, the mammary glands, and the salivary glands. Now, interestingly, the final geometries differ. So the lungs look very different from the kidney, look very different from the mammary gland, and from the salivary gland. But the molecular mechanisms that control the branching are basically the same across organs and across species. We know that fibroblast growth factor is a major player, at least in mice. So here you see lung branching in a normal mouse compared to branching in an FGF knockout mouse. You can see that the branching is severely reduced. Conversely, if FGF signaling is increased in a culture of normal mouse lungs, more branching occurs. Now, there's some evidence that FGFs are important in humans, but these experiments also indicate that FGFs are critical for branching in lung development. And not surprising, they've also been implicated in kidney and salivary gland development. Now, FGF is not the only signal, of course. There are signals that also stop branching, but all of those inductive and inhibitory signals are balanced during development to achieve proper branching. So if you see errors in branching morphogenesis, this results in defects ranging from abnormal numbers of pulmonary lobes, abnormal numbers of bronchial segments, both increased and decreased numbers of lobes and segments. Well, let's recap one last time. We've now formed the trachea, 2 lungs, 5 lobes, and numerous segments, with the pleura and pleural cavities formed. And we've essentially set up the patterning of the adult lungs. In terms of timing, we've now completed the embryonic, the pseudoglandular stages, and we are partway into the canalicular stage. So at this point, around 17 weeks or 17 to 24 weeks, the conducting zone is essentially built. In my next video, I'll take us through the rest of these stages as we see how the gas exchange or respiratory zone is formed. Thanks for stopping by.